So here we're going to talk about how glycogen molecules are actually formed in cells. And so it all begins with a protein that's called glycogenin. And glycogenin is going to initiate a new glycogen molecule to be formed. So glycogenin is a protein, it's an enzyme, and it's also referred to as the primer, the starting place for your glycogen molecules. So being an enzyme, it's going to have many different pockets. So you can see here that our glycogen precursor, UDP glucose, is going to be able to bind to the pockets of your glycogenin. And you're going to have magnesium, which is important, and this is going to provide electron pair acceptor that's going to stabilize the UDP group coming in. So glycogenin has two reactions. It's going to have a tyrosine 194 in its pocket that attacks the hydroxyl group of the anomeric carbon on the actual glucose molecule, and it becomes a glycosylated tyrosine residue. And of course, this is attached to the molecule of glycogenin. The carbon 1 of another UDP glucose is then attached by the carbon 4 hydroxyl group of the terminal glucose, and you repeat. So this is where we're building our alpha 1,4 bonds. So here we have our glycogenin with the tyrosine that's now going to be glycosylated. Here's the glucose attached to tyrosine through that glycosylation. And then you have another UDP glucose. This is going to be a 1,4. So this is going to be the alpha 1,4 link. And then you're going to continue to add. So here's another 4. And then you're going to have another UDP glucose. And that will come in and attach to that. And you'll start to get a chain of glucose added to the glycogen primer. Now the glycogen chain is again going to be elongated by glycogen synthase. So that's the enzyme that does this. And it's simply going to add to the incoming UDP glucose to the non-reducing end of the glycogen molecule. So here we're going to have our non-reducing ends of the glycogen, and here we're going to have a UDP glucose coming in. Glycogen synthase releases UDP, and you add that new glucose molecule, and you produce a new non-reducing end. And over here we have glycogenin. Now, as you add glucose units, they get long. And so in order to have more non-reducing ends to build this molecule, you have to branch. And so the branching enzyme is amylo-4,6 transglycosylase. So basically you're going to make an alpha 4,6 bond. And so we know the structure of glucose, and glucose has that 4 carbon, that's the non-reducing end, and also has that 6 carbon. And so those are going to link to form the 4,6 bond. So here you have your glycogen core, we'll call it, as your book calls it here, and it's this long string of glucose. And then when you get about 11 residues, you're going to transfer 6 to 7 of the glucose residues and form a branch. And so you can see here they've color-coded it. So here's our glycogen core, which contains glycogenin. And then you have your glucose residues, and you don't take the glucose that's going to be close to the branching point. You're actually going to move this, this group of glucose is going to be moved to this branch. Okay? And then you're just going to make the alpha 1,6 branch off of this. Now, your glycogen is going to continue to branch and continue to form long chains off of all these new non-reducing ends. This is all going to take that energy built up in UDP glucose, which will release a free UDP each time. And the transfer is going to be done by glycogen synthase, and then your branching enzyme is going to form that alpha 1,6 branch. Now the molecule overall will have a central glycogenin. So every time your cell wants to make a new glycogen molecule, it has to start with glycogenin as a primer. And then it'll have different tiers. So you'll have your primary tier, second tier, third tier, etc. And you're going to continue to branch to make additional tiers. There's up to 12 tiers on a mature glycogen molecule, which means it'll have about 55,000 glucose residues. Now on the outer tier, these chains are not going to be branched, so these are just simply going to be the non-reducing ends that will be broken down by your phosphorylase enzymes. So glycogen is composed of glycosyl units from UDP glucose. You're going to form the alpha 1,4 glycosidic bonds. You're also going to have alpha 1,6 branches that occur about every 8 to 10 glycosyl units. The synthesis requires that UDP energy, and the transfer from UDP to the non-reducing ends occurs on existing glycogen chains. If you don't have an existing chains, you need to start again with glycogenin.